Okay, so we're the work group on microRNA biomarkers, and we just wanted to give a brief update. Brian and I will be presenting this. Next slide, please. So we have participation from a number of different entities, and we want to thank everybody for all the work so far. And particularly New Cells, who are embarking on our most recent endeavor that Brian will go over. Next slide, please. So the overall goals of this group are to identify either singular microRNAs or microRNA panels that can be used either alone or in conjunction with other biomarkers for nephrotoxicity. We have a broader interest, but whoops.
Great.
Yeah.
Peter, it looks like the newest deck didn't get in, so let me just get the right slides up.
Okay,
So sorry. No, that was my bad.
Oh, no.
Still no?
No, still no.
Those aren't the right ones, or you don't see them?
No, I think that's still not the most recent. So it's okay. Let's just go ahead.
Okay, sorry.
In the interest
Sorry.
of time. No, that's all right. So we want to figure out how, in both a technical way and a theoretical way, how to best implement microRNAs as biomarkers for toxicity, and it has some unique properties that make them particularly suitable for especially translation across species. So the bridging of the preclinical testing to clinical trials is particularly useful. Next slide, please. So we've published a paper where we did a study on a pre-existing data set looking at nephrotoxicity that was detected in non-human primates. And there were urine samples that were collected that we piggybacked onto a traditional biomarker study. Next slide, please. And here's the study design. So the compound is proprietary, but it is an antibody conjugated with a payload of the said nephrotoxic drug. And the nephrotoxicity was first detected by histopathology. And so we wanted to look at early versus mid to late time points in this study to identify which markers could diagnose / predict renal toxicity. Next slide, please. So we were able to make several different correlations. We actually also, if you look toward the bottom half, these are traditional protein biomarkers. And then toward the top, we looked at urine microRNA biomarkers, both in the unfractionated urine or the extracellular vesicle or exosome fraction. And so we had some moderate successes in terms of correlation with histopathology, of course, and especially in the intermediate weeks, but it was not really constantly there was no singular biomarker that was able to perform well over all of the time points. Next slide, please. So we did identify some microRNAs that were potentially useful, although not as singular biomarkers. There were slightly, by using a subfraction of the urine, the extracellular vesicles, we found that those actually improved performances in some ways. And so there may be some mechanistic insights that can be gained through that. And then but the study was underpowered, and so these are really just hypothesis generating experiments that hopefully will be expanded upon in future studies. And so I'm going to hand it off to Brian to talk about our follow-up study.
Yeah, thanks, Peter. So a lot of the work that we've done with the sort of the microRNA panel and development for nephrotoxicity has been done in vivo, looking in rat and, as Peter just described, in monkey. And we wanted to take some of what we learned there and some of the candidates in vitro to, one, see if we see a similar type of release, and to see how they would react in sort of this context. And so we partnered with New Cells that has a primary proximal tubule model that we could test our microRNA biomarkers in. So next slide, kind of show that. So really the overall goal of this was to help develop our microRNA panel, but also with the benefit of helping develop the in vitro system of nephrotoxicity. In other words, if we can see these specified markers in this model, we can basically develop assays that could help support this type of system. All right, one click more. And along these lines, we wanted to determine how much of the media we would need to collect from these in vitro systems to measure the microRNA and also determine if there's a response to nephrotoxin exposure. And so we decided to test this systematically. All right, next slide. A couple clicks too, Connie. Sorry, I probably should have done all at once here. Yep, back one. So these cells are in a transwell format, 24-well plate. We designed the pilot to have an N of 4 for each of our conditions, and we had a number of those. So we used two well-known nephrotoxicants, cisplatin and polymyxin B, at concentrations which we would know we would elicit a response, and their matched vehicle controls. Because we were not sure of the dynamics of the release, we looked at two separate time points, as well as the location of the release itself, to see how they would be similar or different. So we took measurements from both the apical and the basal lateral side of the transwell, in addition to the cell lysates themselves, to see how that might alter the intracellular amounts of microRNA. Okay, next slide. So the targets we looked at are listed here. A number of these targets were derived from what we saw as positive hits and specificity to the proximal tubule cells in our rat study, and that includes the microRNAs like 21 and 192 and 34C. We also performed a technical control. In this case, we used 159A as a spike in control to see how that might vary due to technical factors. In terms of the methods, the measurements were done by TagMan PCR, and you can kind of see the regimen we followed to perform those measurements. Again, we were trying to optimize how much media we needed here, so we took a range of media input, performed the spike in, isolated using standardized methods, treated with DNase, and then did a targeted reverse transcriptase reaction. And I'll show actually both single and a multiplex reaction. We did not do a preamp, and then again followed up the measurements with TagMan PCR. Okay, next slide. So we performed the experiment, and really the first study we did was to optimize the protocol. So I mentioned we ended up doing a single plex. Here we did both single and multiplex in pooled samples using a pooled cisplatin apical sample, which we anticipated there being high amounts of microRNA release. We went ahead and maximized the amount of input, used 200 microliters of input, and looked at miR192, which was highly expressed in the proximal tubule line, and also anticipated a large amount of release into the media. In the multiplex, we used an 8 primer set looking at all the microRNA. So what I'm showing here are the amplification curves from that study. Looking at, on the very left, the amplification curve of a single plex miR192. So in other words, that was the only thing that was targeted to be measured. And then on the right, you can see the plexed 192. So the RT was performed for all of the specific microRNA targets, and that these are the same amounts of input. And so you can see that shift. So there was less amplification of that 192 because of the dynamics of the multiplexing. This also occurred with other targets such as 34A, which you can kind of see as lower expressed compared to 192. But also sort of right shifted based on single plex. So the decision was made, even though it was a little bit more expensive, a little more low throughput, to go with the higher sensitivity of the single plex reaction. Okay, next slide. Yep, and I said all that. So, sorry. Okay, the second study, will this work for all the microRNA targets? So we focused on 192 here. We went ahead and took a step back and did a single plex with all the targets using our two highest amount of tested inputs of 200 and 100 microliters of media. And again, looked at the pooled cisplatin sample from the apical side of the transwell. So next slide shows sort of the results of that. We were able to detect all of the targets, and here you can get an idea of what's more highly released into the media by proportion. And so miR21 was one of our higher expressed microRNA in the apical side, followed by 192, 29C, and 34A. 48, 6, and 34C were lowly expressed. We kind of anticipated that. And we had to actually use higher amounts here just to get a register here. And if you hit one more button, I think that's the conclusions there. Yeah. Okay, next slide. So we wanted to optimize the amount of media input. That was really one of our primary goals of doing this pilot study. In other words, what could we get away with? The important thing here was that if we could use less amounts of media, we could use the other parts, the rest of the volume on, for example, protein biomarkers, which we can help support the study. So we took, again, cisplatin and vehicle-controlled pooled samples from the apical side of the transwell and looked at 10, 50, 100, and 200 microliters of media input, and again, did a single plex of all of our 8 microRNA targets. Next slide will show the results of that. And these are sort of select of sort of a high and lower expressed microRNA. And what you can begin to see is this pattern of, obviously, as you increase the amount of input, you get a higher signal. However, for some of these microRNAs, such as 34C, you actually get a tailing off. And so what we think is happening here is as you increase the amount of input media, there could be RT inhibitors and other inhibitors of the amplification that sort of inhibit the quantitation of, especially the lower expressed microRNA in the media. If you hit one more slide, you can kind of see a summary of the button of all the microRNA, and that pattern holds true, especially again for the lower expressed microRNA. So things like 34C, 34A, 29C, we hit this sweet spot between 50 and 100 microliters to input, whereas it's not too low where we're not actually seeing any measurement for these lower microRNAs, but not so high as where we begin to see inhibition, and we don't see that same fold change compared to our vehicle controls. So the decision was made to go back to all of our samples that were derived from our pilot N4 and do the individual measurements using 50 microliters of volume, which would really be ideal in terms of, again, conserving volume for other protein measurements from a larger study. Yeah, there's some conclusions there. Okay, next slide. So here are the individual samples showing the standard deviation error bars. This is from the apical side of the transwell. Again, looking at our matched vehicle control, so water to cisplatin and DMF to polymyxin B, and looking at the comparisons here. What sort of most striking here is one, the variability. We see a lot of that, especially on the apical side. But also, when we see a trend or we see a statistical change from vehicle control, they're more than likely in the higher expressed microRNA. So 21, 192, 34A, we're all in the higher end of the spectrum compared to 29C, 46, and 34C. And this is looking at 2 time points too, so 24 hours and 72 hours after exposure. And you can see in some cases that does affect the dynamics, in others it doesn't really. And we can kind of see the same kind of trends. Even more interesting is the next slide, the basolateral measurements. Again, a decent amount of variation according to our standard deviation error bars here. But what's interesting here is that baseline for a lot of these measures, we don't really get much measure of these microRNAs, with some of the exceptions like 192. But when there's some sort of perturbation, when there's a nephrotoxicant exposure, we see what's basically going from nothing to something. And that really enhances our ability to detect an event, a toxicity event. And so this is where we really begin to see statistical changes, again, for these higher expressed microRNAs. We had more trouble with these lower expressed microRNAs, a lot more variability. And that does tend to indicate that we, at least in a larger study, may be pushing the input volume maybe a little bit more to get this type of detection. Okay, next slide. Go ahead and click through all of these, Connie. I'll just go ahead and summarize this. So we chose the single plex RT, and lower throughput, but gave us a higher detection rate. We concluded that between 50 and 100 microliters of media input really gave us our best chance to detect most of these microRNA, and our ability to look for increases due to nephrotoxicity in these cells. Despite the variability, we could see these changes to these nephrotoxicants, and again, especially with these higher expressed microRNAs. And the basolateral was most sensitive because of really the detection of the baseline when really nothing there to something when nephrotoxicant was present. Okay, and one more slide, I think, for me. And the spiking, sorry, I didn't mention the spiking levels in intracellular microRNAs really didn't change, so we didn't see a technical bias, and really not much change in the intracellular microRNAs, despite this toxicity and release. It was pretty consistent, so not a lot of cell loss, but we did see release these microRNAs, so an indication that it's maybe even more sensitive. Okay, next slide. And so our follow-up is to do the full study. The same compounds, but we want to do more doses, technical reps using a different biological or donors in the cell culture, add one more time point, and again, look at the basolateral and apical side of the membrane. We will take these measures from the same well for each of these conditions. So collect media and replace at 24, 48, and 72 hours. This conserves on the wells that we're using, and also gives more of a well-to-well match to our exposure regimen. And then the cells themselves, we will do some imaging and some toxicity measures, and perhaps also the microRNA, the intracellular microRNA measures at the end. And so we should get a nice full data set with some multiple doses and time points that tell us really the dynamics of this release and how sensitive this assay may be with these microRNA biomarkers. Okay, I think that's all I have. I'm going to turn it back to Peter to talk about future directions of the team.
Yeah. All right. All right. So we want to actually develop from this study, as well as building on our previous studies. There was a study looking at laser capture microscopy and looking at segment-specific markers, as well as segment-specific nephrotoxins, and to leverage all of that, looking at some mechanisms in terms of being able to understand what the role is of these microRNAs, and ultimately the idea would be to get to move toward qualification. with or without combining with traditional biomarkers. And go ahead, click again, please. And while we've discussed some other ideas, we could look at other small RNAs such as tRNAs, Pee-wee RNAs, and then looking at other tissue and cell types such as cardiotoxicity and hepatotoxicity. So, however, this has sort of been our focus, and I think one of the things that we're looking in terms of mechanism is this idea of polarity. Now, the new cells system is constantly under continuous improvement. So one of the things that may be in the future is looking at flow versus non-flow. You don't really have cilia formation in the non-flow, but you start to see cilia formation with the flow. So that may be actually important in terms of the functioning and response of these cells in culture. And then finally, we would expect that the release of microRNAs into the apical side versus the basolateral side would reflect more a urine biomarker, whereas the basolateral side would be more of a plasma biomarker, circulating biomarker. So I think, yes. So here, so we finished our EV study, and we were wanting to look at the in vitro work. This is initially with the proximal tubule cell model that is very well developed. However, in development is multicellular in vitro system that maybe includes, say, distal tubule cells, as well as perhaps a separate one would be looking at the 3 different cell types in the glomerulus. And of course, we want to narrow down what kind of panel we can develop from the microRNAs. And we are looking for additional ideas, as well as anyone who wants to join us. Thank you.
Great. Thanks so much, Peter and Brian, for that talk. We have a few minutes for questions, if anyone has any. I think kicking off, one thing we've talked about a little bit in the group, but maybe Brian or Peter could speak to, is a lot of what we've done is PCR to measure the microRNAs. Maybe, Brian, you could speak to a little bit what's maybe on the horizon with sequencing, and when that might be applicable for us, or whether we stick with PCR.
Yeah, in the rat study, we did employ a lot of different types of measures that could be conducive to current and future work. One of the assays we really liked was the Abcam Fireplex assay, which they did discontinue. The nice thing about that, it was sort of a we didn't have to isolate the small RNA to do the measures, so we could do direct measures regardless of the input. So it could be cell lysate, it could be urine, it could be blood, what have you, and we could do those measures. So a nice, I think, future sustainable assay would be something along those lines. And actually, TempoSeq is coming out with a microRNA assay. So in other words, it's a small RNA back into their established TempoSeq technology. And they also have sort of a direct measure protocol, which could be employed here. And so that's something, at least my lab has begun to look into and could be maybe utilized in the work group studies. In terms of discovery, you can still use things like small RNA sequencing, which we do quite a bit. Nanostring, you could do that too. That's a nice assay that you can use in conjunction with both mRNA and microRNA measures. And even along the PCR realm, digital drop PCR is a really sensitive measure, which we've used as well. We didn't use it here in the vitro study, but it's something we could also utilize in the future. So a lot of different options and a lot of different possibilities for sure.
Thanks, Brian. Any other questions, comments from the team?
Brian, I have a quick technical question. The 50 microliter you're talking about, is that per RNA sample, per miR sample? Is that correct? So if you have A, that will be 400 microliter?
Actually, it's for all of them. And so we did, we took that, isolated the small RNA, and then did all the measures on that. So we get plenty of material to not only run all 8 of our microRNA, but beyond that. To be honest, I could look up what the yield is, but it's enough to do the measure. So yeah.
So one isolation, just PCR separate, correct? Right?
Correct. Yes.
Okay. Thank you.
Thanks, Yuen. Okay, I think we can thank Peter and Brian. Thanks so much for walking through the microRNA progress. A lot of exciting things to keep an eye out for next year. Thanks both.
Thanks.